This next set of videos is going to focus on the descendants of primary endosymbiosis that led to plants. So looking at the acquisition of chloroplasts and who came after that. So if we're thinking about what a protist isn't really anything because it's just a eukaryote that is not a land plant, an animal, or a fungus. And so if we look at what those are represented as here, here's animals, here's fungi, here's plants. All of these other groups are all protists that used to be in the same kingdom, Protista, but now we know they're not related. So we have looked at oomycetes here in the Straminopilia. We're going to look at their descendants in a following lecture, but here we are going to be looking at the red algae and the green algae and a little bit of the glaucophytes. So we're looking in this group, the Archaeplastida. Just a little interesting point, this group here, the Apicocomplexa, it's a diverse group of parasites. They all have plastids still, but they're no longer photosynthetic. So the disease that causes malaria and toxoplasmosis, both of those are in this group. Weird. Okay, so a little overview. First, we're going to review primary endosymbiosis, and that'll be this video, and we'll look at the glaucophytes. And the next two videos we'll cover separately the red algae and the green algae. So review of primary endosymbiosis, we had our blue-green algae, right, our cyanobacteria that are not algae at all, and they have two membranes. So this is our cyanobacterium, or cyanobacterial ancestor. And it has a plasma membrane and it has a cell wall. And that outer cell wall has peptidoglycan in it. Because it's a bacterium. So this bacterium gets ingested, but not digested, by a heterotrophic eukaryote. So here it is, it's a eukaryote, there's its nucleus, and it is eating our cyanobacterium. And it engulfs it. Here's our nucleus again with our nucleolus. And so here's the membrane that engulfs it, but it doesn't get digested, it stays inside there. And eventually it's going to become a functional organelle inside that eukaryote. And so that is endosymbiosis, this idea of having two things that are living together, that symbiosis, but one is physically inside of the other one. So this cyanobacterial ancestor is going to, over the course of evolutionary history, become a chloroplast. And it'll have two membranes. One of these membranes is going to go away, so it'll just be left with that membrane of the thing that engulfed it, and then its original membrane. So we can review that process in a few different stages. So here we have our heterotrophic eukaryote, and it's engulfing our photosynthetic prokaryote. So that's our cyanobacterial ancestor. Gets engulfed as this thing tries to eat it and totally surrounds it. That cyanobacterium doesn't get digested, it just stays alive inside that host cell, and it's photosynthesizing, so it's making sugars, and those sugars can maybe leak out into the host cell and allow that host to survive and reproduce better because it doesn't need to spend as much time trying to get food. It has food in the form of sugar coming from its future organelle. So there's two membranes on these plastids. The outer membrane came from the original host that engulfed it, and the inner membrane comes from the cyanobacterium. The cyanobacterial cell wall eventually goes away, but we'll see this possible transitional phase where it's still there. And that possible transitional phase exists in the glaucophytes. So it's really not resolved who came out first in the evolutionary tree of life with the algae and early plants, but we can make some guesses at how that happened. And in a lot of people's guesses, it's the glaucophytes that come out first because they have a lot of ancestral characteristics. So here we can see that they have these two large plastid structures inside them. And those two large plastid structures are called cyanelles, And they're called cyanelles because they're not quite chloroplasts yet. They're very similar to chloroplasts, but they have a few distinct differences that are more similar to the way that cyanobacteria function. And one of the differences that is more applicable for our class is that there's still some remnant peptidoglycan between the two membranes of the cyanelle. And so that gives us some evidence that that cyanelle has a bacterial ancestry. So remnant peptidoglycan, whenever you see peptidoglycan, you're thinking cell walls of bacteria.